Today, we're jumping into a really cool study that basically shows how your everyday consumer-grade camera can become a serious scientific tool for understanding plant health. We're talking about getting powerful data that can actually compete with some way more expensive equipment. So here's what we're going to cover. First, we'll talk about why high-tech phenotyping can be such a headache. Then we'll look at the super accessible camera-based solution they came up with in this study. From there, we'll dive into the brand new insights this whole method unlocks. We'll also get into the weeds a bit on the tricky puzzle of image correction, and then we'll wrap it all up with some clear takeaways for researchers and managers. All right, so first things first, why do we even need a new approach? Well, it really comes down to a huge barrier to entry when you want to do any kind of advanced plant analysis. And the authors really nail it with this quote. Top-of-the-line research usually means a massive investment. We're talking about these complex facilities, super pricey sensors, and you need a whole team of specialists just to run the thing. This basically puts all that powerful analysis completely out of reach for a lot of people. Students, managers, even well-funded researchers. And that brings us to the really clever solution in this paper. The big idea here is that you can get legitimate scientific-grade data using tech that's affordable and accessible to just about anyone. Let's see how they pulled it off. Okay, the setup is just brilliantly simple. They took a consumer camera, nothing fancy, under $1,000, and put it inside a custom do-it-yourself light box. But here's the magic trick. It's all about control. They locked in all the manual exposure settings, ISO, aperture, shutter speed, and then, this is crucial, they set a custom white balance for their specific lights. And this right here is the fundamental principle. It's not about having the most expensive camera money can buy, it's about creating a repeatable, totally consistent environment. When you lock those settings and control the light, every single photo becomes a standardized measurement. You've essentially turned a simple camera into a reliable scientific instrument. So the setup is straightforward, but what can it actually tell us? This is where the study gets really powerful, because it starts to look for signals in the data that go way beyond just how green a plant is. For years and years, the go-to approach in this field has been to just measure the percentage of green pixels in a picture. And look, it's a useful metric, no doubt about it, but it's only telling a tiny part of the story. And this is the big question, right? If our only tool is measuring green, what other signs of plant health or stress are we just completely blind to? Well, it turns out we're missing a whole lot. The first big breakthrough here is the idea of quantifying the yellow area, what they call %Y. So instead of just throwing out any plant material that isn't green, this metric specifically measures the percentage of yellowing tissue, which of course is a direct visual cue for stress, aging, or some kind of deficiency. Next up, the authors created a totally new index they call HSVi. Now, this is more than just color. It combines hue, saturation, and value to pick up on really subtle information that simple color metrics miss. Think about the richness of a color versus just its brightness. That can tell you a lot about changes in vegetation density or different kinds of stress. And they developed another new one, the BA ratio. This one is clever. It uses the C-Lab color space, which is designed to be closer to how humans actually see color. And this ratio zeroes in on the balance between blue-yellow and green-red, giving us a totally new way to see those shifts in pigment that signal a plant's health is changing. So are these new metrics just academic, or are they actually useful? Oh yeah, they're useful. This slide right here is the payoff. That simple % yellow metric had an incredibly strong correlation of -0.89 with visual ratings from human experts. In plain English, as the amount of yellow went up, the expert quality rating went down, almost in perfect lockstep. That is a massive validation. And to put that into perspective, just look at this chart. The new yellow area metric could explain 79% of the difference in those expert readings. A standard metric, the dark green color index, only explained 62%. So what's the key takeaway? By just measuring yellow, we get a much better idea of what a trained expert sees than by only looking at green. Now, for anyone who's actually worked with this kind of data, you know it's not always this simple. So the authors also did a deep dive into the nitty-gritty of image processing in a supplementary document, and that adds some really important context here. The million-dollar question is always, do you correct your images or not? Applying things like color correction seems like a no-brainer. It should make things more accurate. But as you can see here, it's a real trade-off. Corrections add a ton of complexity, and they can even create new, subtle errors that are really hard to track down. Sometimes the simplest path really is the most reliable. That deep dive had some incredibly practical advice. For example, a high-quality JPEG can work in a pinch, but a lossless format like TIFF or RAW gives you way more flexibility later. Better lighting, better lens settings, like shooting at f/8, that can all improve your raw data. But here was the biggest takeaway. The effects of corrections were all over the place. Something that improved one metric might do nothing or even make another one worse. Okay, so after all that, what are the practical, actionable things you can do with this information? This last section really boils it all down into clear guidance for anyone who wants to apply these ideas.
This is probably my favorite slide in the whole deck. Look at how well these super simple camera-based metrics line up with established complex measurements. Percent green is basically a stand-in for NDVI. And the new ones? BA ratio correlates at 0.90 with NDRE. And HSI has a 0.90 correlation with near-infrared. This proves that these new, simple indices are capturing real, meaningful biological signals. It's fantastic. So, let's wrap this up. Here are the main things to remember. First, embrace simplicity. A regular camera is a beast if you control the environment. That means locking your manual settings and always setting a custom white balance. Second, stop looking only at green. Start measuring yellow. It's an incredible proxy for visual quality. And finally, be really careful with corrections. Test them. Their value depends entirely on your specific research question, so you have to see if they're actually helping. And that really brings us to the final thought here. What this research does is it makes powerful plan analysis accessible to everyone. It proves that you don't always need the fanciest tools to find profound insights. Sometimes it's just about being creative and really controlling your methods. Which leaves us with a pretty exciting question, doesn't it? What's waiting to be discovered in your next dataset?